Yes, good morning, students. Good morning, everyone. Am I audible to all?
Yes, sir. Good morning.
Yes, sir.
So as you can see that our today's topic, the complex permanent tissue, previous lecture in which we had already completed about the study of xylem. So we shall be focusing upon the study of phloem now. Phloem tissue. See, basically phloem tissue is meant for transportation of organic food primarily. Mainly, it is supposed to transport the organic food. Normally, the photosynthesis performed by the leaves, the organic food produced by the leaves, transported towards the different parts of the plant body. That's a normal trend. Sometimes the food that is being stored in the root region, it has to come up towards the emerging leaves in certain season. So phloem is the one actually that permits transportation of food or materials from leaves to the root region and even from root region to the leaves region. The bidirectional movement can be seen through this phloem tissue. Normally, the xylem is performing unidirectional transportation root to the leaves. Xylem is performing this unidirectional transport, whereas phloem is performing bidirectional transport. That one needs to understand. If we see the components of this tissue, in case of the two different types of plants, two major groups of plants, we can say, then it will be like this. Angiospermic phloem, the more evolved plants, the angiosperms. Their phloem, it is carrying, sieve tube elements, companion cells, Phloem Parenchyma, and Phloem Fibers. These are the constituents present in the angiospermic phloem. I repeat, sieve tube elements, companion cells, Phloem Parenchyma, and Phloem Fibers. Actually, the main conductivity is carried out by this sieve tube elements, helped or assisted by companion cells, whereas parenchyma and sclerenchyma fibers, which are part of phloem, but for some other additional functions. So they are also part of phloem only. On the other hand, we have a gymnospermic phloem, the phloem tissue in the gymnospermic plants. It includes sieve cells as a conducting elements, and to assist them, to help them, supporting cells are albuminous cells, or even you may call it as the Strasburger cells, equivalent to the companion cells of angiosperms. Here you can see that sieve tubes are not there. So it's a primitive type of a phloem that is to be found in gymnosperms and pteridophytes. Here, instead of companion cells, these are known as albuminous cells. So these kind of variations prevail in the components of phloem as far as gymnosperms and angiosperms are concerned. Even in xylem, you have seen sieve cells to be found in gymnosperms, sieve tubes to be found in angiosperms over there, mainly as a conducting elements for xylem. So basically, gymnosperm is comparatively in evolution a primitive plant. So its xylem phloem have primitive components. Same manner, जैसे xylem में trachid और phloem में sieve cell, gymnosperms में. Similarly, angiosperms are a bit more evolved. They have a bit higher level components, such as in xylem they have a vessels, in phloem they have a sieve tube. We go ahead. One by one, we shall study about these components, starting with the first one, sieve tube elements. We will focus upon angiospermic phloem. So we shall be discussing about sieve tube elements, companion cells, phloem parenchyma, and phloem fibers. Let's begin with the first one, sieve tube elements. Clarity is an issue. First of all, let me show the gymnosperms, their constituents of phloem, mainly the sieve cells and albuminous cells. Sieve cells and albuminous cells belonging to gymnospermic phloem. On the other hand, here is the phloem in case of angiosperms. Look, the sieve tubes, companion cells, phloem parenchyma, and there will be parallelly running those sclerenchyma fibers, which will be known as Phloem Fibers. So sieve tube element, companion cell element, phloem parenchyma element, and last is phloem fibers. These kind of set of constituents make up the cylinder, that is the phloem tissue inside the plant body. Beginning with the first in the sequence, and that is sieve tube element. These are generally long tube-like cylindrical structures arranged longitudinally one above the other, which means this is one sieve tube element, another sieve tube element above it, another one above it. So inside the plant body, you can see that series of sieve tube elements placed one above the other make a sieve tube. Individually, this one is a sieve tube element. जैसे था ना वो vessel members placed one above the other and transverse walls degenerated to make a continuous vessel. Similarly, sieve tube elements placed one above the other to make a sieve tube. Now, if you look at their transverse walls for the sieve tube elements placed one above the other, let's assume that this is the one. Another one placed below it. So in this region, they have perforations. So when they get placed one above the other, materials can be conducted via this perforated transverse walls. These perforated transverse walls They are called as sieve plates. They are known as sieve plates. These perforated end walls of these sieve tube elements, they are sieve-like structures. They are known as sieve plates. It's via these sieve plates conduction of organic food in the form of protoplasmic strands. The material is transported from one component to another one. That is how in the sieve tube the transportation will take place. Look at this individual one. Have a look at this. This will give you an idea what is a sieve plate. Let us assume that this is a one sieve tube element. It is placed above the other sieve tube element like this. At the juncture position, the transverse wall between them, ये एक सेल, ये दूसरा सेल, so these position where these end walls of these two cells, cell number 1 and 2 are meeting, in that position, as you can see, there are lots of apertures. It's highly perforated. It is highly perforated. These perforated plate is called as sieve plate. Via this plate, materials will be transported from one into another one. I hope you get the idea. Is it getting clear to you? Sieve plate within the long sieve tube, at every two sieve tube elements meeting each other, the end walls with the perforation, these end walls being known as sieve plates, the pores being known as sieve pores. It is via these pores material is transported from one sieve tube component to another sieve tube component. Like that the transfer occurs. थोड़ा और better view अगर लेना है इस matter का, look at this one. This is the one. I hope now you must have got the sense of what I'm trying to explain to you. The sieve plates. Clear? Every sieve tube member at its end walls having perforation known as sieve plates, pores called sieve pores, via which from one sieve tube member into another sieve tube member the protoplasmic strands are passing, and in that format only the material is transported from one cell to another, and within that sieve tube transport occurs. As you can see that these cells are lacking nucleus, sieve tube cells, sieve tube members rather. Although they are living cells, they have the protoplasm in them, but they are devoid of the nucleus. Actually, their activities are governed by the nucleus of adjacent cell known as companion cell. Known as companion cell. As you can see, it has its nucleus. So the nucleus of adjacent companion cell controls the activity within which cell, within the adjacent sieve tube member or sieve tube component over there, sieve tube cell over there adjacent to it. You see they have a plasmodesmatal connectivity. I am sure you people understand what is plasmodesmata. Do you understand what are plasmodesmata?
Yes, sir. Cytoplasmic strands through which
Adjacent
cytoplasm of adjacent cytoplasm come in contact.
connected. So these are basically connections between the two adjacent plant cells where their cytoplasms remain interconnected, you can look at those plasmodesmatal connectivity. Is it seen? I have enlarged it. Between the companion cell and the sieve tube member. Can you make out this?
Yes, sir.
Yeah, so that is the point which you need to understand, that this is how the nucleus of this cell is able to control the activity of this cell, because that one is without nucleus. The sister cell, also known as companion cell, also known as companion cell. It is basically a parenchymatous cell. By nature, it is a parenchyma cell, a living cell, one which is having the nucleus. It is placed adjacent to the sieve tube element. This both will remain connected via pits, pits in their walls. Pits मैंने बोला था apertures होते हैं. And via these pits, cytoplasm of one cell can go into the other cell to regulate the activities. So both are connected by the pit fields. Pit fields means एक area जहाँ पे companion cell को भी pits होते हैं और exactly parallelly on the other side sieve tube member को भी pits होते हैं. दोनों side पे pits available होते हैं. ऐसा. So pits to be found on both sides, parallelly arranged, get arranged, and they together they together become the area where the transportation of materials can take place, mainly from companion cell into this sieve tube member, and regulate the activity. Materials which go from here to here, mainly the sugars, इसके adjacent photosynthetic cells होते हैं. So from there, the sugars are loaded into this cell, companion cell, and from companion cell, sugars are loaded into sieve tube members, and then they are transported towards the root and other regions over there. That's how the whole activity occurs. In this process, the sugars, when they come into these cells, which cells? Sieve tube components. Concentration of the contents of this phloem, that is called phloem sap material, its concentration increases. So we say the companion cells have helped in maintaining the pressure gradient in the sieve tube. अब ये क्या है पूरा pressure gradient, हम 11th chapter में learn करेंगे. I have to show you that in 11th chapter you have one such topic. ये वाला, अरे बाप रे गड़बड़ हो गई. ये वाला, have a look. Sugars are loaded into this phloem via companion cells, as you can see, via companion cells. जैसे photosynthetic cell है, फिर वहां से companion cell, फिर वहां से phloem में आता है. So जब ये sugar phloem में आता है, so adjacent xylem में से water आता है, water is coming into this phloem, and together water and sugar, they increase pressure over here. Because of that pressure, the sugars and other materials will be transported towards the other parts, stem and root. So this is how the phloem transports its material. In this entire activity, companion cell played a significant role in bringing the sugar in, increasing the concentration of the phloem sap, and maintaining the pressure gradient in this zone, in this particular zone. So that is the role of companion cell I had to explain. See, getting the sense of what I'm explaining, sieve tubes and companion cells up to this point, is the thing clear to you?
Yes, sir.
A quick recap about the sieve tube and the companion cells. Sieve tube elements are long tubes made up of many units known as sieve tube members placed one above the other, having their end walls meeting each other, being perforated. These end walls known as sieve plates, the openings in them known as sieve pores. Through these sieve pores, materials get transported from one sieve tube member into the other one in the form of protoplasmic strands, and that is how the transportation takes place in sieve tubes. The components of the sieve tubes known as sieve tube members, they are without nucleus, enucleated. Their activities need to be controlled. For that, the adjacent cells known as companion cells are useful. It is these companion cells having the nucleus in each of them, the parenchymatous living companion cells adjacent to the sieve tube component. You can see that laterally the walls of the tube are in contact of each other. Now, in these lateral walls, they have specialized areas which are known as pit fields, where pits, the openings in the walls, cell walls, present from both sides are parallelly arranged. So pores of both sides are arranged in such a way that cytoplasm from the companion cell can pass into the sieve tube element. ये एक sieve tube नहीं है, ये एक sieve tube element है. बहुत सारे sieve tube elements एक दूसरे के ऊपर अरेंज होके sieve tube फॉर्म करेंगे. This arrangement will help in maintaining the pressure gradient in the case of sieve tube, due to which transportation will occur in the sieve tube. Additionally, you can see that there are some parenchymatous cells on outer side, particularly outer to the companion cells, not in direct contact with the sieve tube, but they are outer to the companion cells. These elongated, tapering, cylindrical parenchymatous cells, they are called as Phloem Parenchyma. They are known as Phloem Parenchyma, each with a dense cytoplasm and nucleus. They have a very dense cytoplasm and nucleus in them. Again, their cell walls have a pits. Wherever these pits are present, again, that is going to help in plasmodesmatal connectivity. Then these cells are meant for storage of material. Do you remember the Xylem parenchyma stores which materials? याद है आपको?
Starch or fats and substances like tannins.
Starch, lipid, tannin, etc. If you can recall those things, isn't it? Here the materials getting stored in phloem parenchyma are different. They are resins. They are resins mainly, latex, latex material, even mucilage. So these type of materials can be stored in the phloem parenchyma. Okay. Generally, this is present in a angiospermic phloem, and in that also mainly in the dicots. normally it tends to remain in a monocot type of angiosperms. Monocots and dicots both are angiosperms. बट डाइकोट का फ्लोएम देखो तो फ्लोएम का पैरेनकाइमा मिलता है. मोनोकोट के फ्लोएम पैरेनकाइमा में मोनोकोट में फ्लोएम पैरेनकाइमा मिसिंग होता है in their phloem. So that is the detailed information on phloem parenchyma. Again, I would go through that. Living parenchymatous cells, elongated, tapering in shape, cylindrical in nature, with a dense cytoplasm and nucleus, present on outer side to the companion cells. They also possess the pits in their cell walls, via which they can also communicate with each other and carry out plasmodesmatal connectivity, and transport of material is possible via them. They normally store materials such as resin, mucilage, latex, etc. Excuse me. Normally they are found in the angiospermic phloem, but in dicots, not in monocots. Last, that is the Xylem Fibers. Basically, they are supposed to be what? Basically, they are supposed to be those dead sclerenchyma fibers to be found in this adjacent to this this rather parenchyma cells, normally outermost in position. These are also known as Mast Fibers. So called Phloem Fibers or Mast Fibers. They are elongated, unbranched sclerenchymatous cells with the pointed needle-like apices. Each one's end, if you see, is sharply pointed like a needle. So needle-like apices or ends they are having. They are thick-walled. They are dead components without protoplasm, you know very well. They are, important in economic importance is there. Many of the plants provide their phloem fibers, which are important economically. Example, phloem fibers of jute पता है आपको? Bengal or Bangladesh are the main exporters of jute. Then flax and hemp. They are commercially utilizable. You know now very well this thing. The bags which are jute bags which are prepared, in which the cereals and all this wheat etc. they are packed over there. So jute bags के लिए ये fibers utilizable है. Jute के, फिर flax plant के, फिर hemp plant के, इनके phloem fibers का economic value होता है. Again, phloem fibers are present in a secondary phloem, not in a primary phloem. आपको पता है ना, जब primary growth होता है उस टाइम पे जो भी tissues होती है उसको primary tissue कहते हैं. So जब plant maturity की ओर जाता है तब तक primary growth के period में जितना भी phloem form होता है उसको phloem fibers कहते हैं. Phloem primary phloem कहते हैं. So that primary phloem is without phloem fibers. जब plant में secondary growth होता है mainly in dicots, so that time it will be having phloem being added, that is a secondary phloem. Such secondary phloem possesses phloem fibers. So ये component किसी किसी में present है, किसी में absent है. ऐसा understanding आपका होना चाहिए. So you could study about sieve tube elements, and then you have also heard about sieve cell, and now sieve fiber. Fiber, sclerenchymatous, dead without protoplasm, very thick-walled, pointed, elongated cylindrical structures, unbranched in nature, normally economically important. Bast fibers are obtained from jute, flax, even hemp plant. normally absent in primary phloem and present in secondary phloem. That is about it. Is this clear?
Yes, sir. Clear.
Yes, sir.
आगे जाते हैं फिर. If you just try and compare the xylem and phloem arrangements. First of all, xylem is meant for water and mineral conduction, whereas phloem is meant for organic material transportation. Okay. Another thing, xylem conducts the material one way only, that is from root to the leaves. Phloem is carrying out this conduction two-way, most of the time from the leaves to the roots. Sometimes stored food from the root can be brought up to the emerging leaves, so it can be even upward. So bidirectional transportation through phloem is a reality, possibility and reality. Here if you see in a vessel, the vessel members, end plates, these regions completely degenerated. So a continuous column is created over here for upward conduction, for conductivity rather, you can say. If you focus upon this phloem part, the end walls are not fully degenerated in this sieve tube members. यहाँ पे जो वेसल मेंबर्स थे उनके बीच में पूरा degenerate कर दिया wall and continuous passages created. Here what is done is, as you can see, end plates are having perforations through which protoplasm will be moving from one to another one. Simple as that. Last, most of the components of the xylem are dead. Only parenchyma, the outermost arranged parenchyma cells, they are, outer cells are not living, what they are, the cells are living but needs. See, bottom line, this point is not appropriate over here. What I would like to explain is this way. Most of the constituents of xylem are dead. Trachea, vessel, fibers. Only living components are parenchymatous tissue, the xylem parenchyma. Reverse, in phloem, most of the constituents are living. Sieve tube members, companion cells, phloem parenchyma. The only dead components are which one? Mast fibers, sieve fibers, you can say sclerenchymatous fibers or phloem fibers rather, not sieve fibers, phloem fibers or sclerenchymatous fibers. They are the dead components over here. This was a brief comparison also between xylem and phloem. I hope you have understood the same. Now, if you have any doubt, you raise your point. I will describe things again. A repetition, if required, we can go for it. If not, then we can move on to our next topic. Sharon, but you have raised hand, you can ask the question.
Yes, sir. Sir, it is about parenchyma. You told that they are only found in angiosperms. Then the next point, it is about monocot, dicot. I didn't get it.
So angiosperms include dicots and monocots. So yes, it is present in angiosperms, but specifically speaking, amongst the angiosperms, it is present in only dicot plant phloem, not in monocot plant phloem. ऐसा दोनों, वैसे तो डाइकोट मोनोकोट दोनों को कंबाइनली क्या कहते हैं? एंजियोस्पर्म. तो अगर कुछ चीज डाइकोट में है तो भी हम बोल सकते हैं द थिंग इज देयर इन एंजियोस्पर्म. कोई चीज अगर मोनोकॉट में तो भी बोल सकते हैं the thing is there in angiosperm. So we generalize that the material is to be found in the angiospermic plants. Further specification. Okay, amongst the angiosperms, this material, the Phloem Parenchyma, is present in their dicot members and it is absent in their monocot members. That's how we take it.
Okay, sir. Thank you, sir.
Yeah. Anyone else with any question? Then we can. Okay, it seems you have no other question left. No one wants to repeat also. So we have concluded the complex permanent tissue. Regarding their origin and development, there is one topic which we have to see after a break. So let's have a break for 5 minutes and then we resume our activity. Madam, stop the recording.
Yes, sir.
Somebody said, sir, who is there?
સર શ્રેયા
બોલીએ.
સર મંડેના છે ને 9 ચેપ્ટર નો ટેસ્ટ છે તો સર એના પેલા રેકોર્ડેડ વિડિયોઝ તમે મોકલશો
મંડે
એ
કયું ચેપ્ટર?
એ પેલું આ મંડેના ચેપ્ટર 9 બાયોમોલિક્યુલ્સ નું છે.
હા મેડમ બિંદિયા મેમ
Yes, sir.
ચેપ્ટર 9 ના વિડિયોઝ ને અવેલેબલ કરાવી દેજો મારા લોકોને.
Okay, sir.
Thank you. All of you have a look.
થેન્ક યુ સર.
Just see this one. One concept I am explaining to you. Understand this. When plant is growing, it will show two types of growths. One is a primary growth and another one is a secondary growth. One is a primary growth and another one is a secondary growth. You know the difference between them, so I'm not going in detail that since sapling stage is still the maturity, it will be having a primary growth, and after achieving maturity, then also it will continue to grow. It is called as a secondary growth. Normally it takes place in dicot plants, not in monocot. Monocot में सिर्फ primary, dicot में primary के बाद secondary growth भी होता है. Always exceptions are there on either sides. Whenever the tissues are formed during primary growth, they are called as primary tissues. Same way, when the growth occurs during this, or rather the secondary growth, the tissues that are formed would be known as secondary tissues. And then you can understand that one of the tissue formed would be xylem and phloem. So there will be primary xylem and phloem. Similarly, on the other hand, we will have a secondary xylem and phloem. बोलो, ये क्लियर हुआ आपको? हाँ या ना?
યસ.
Is it clear to all of you?
Yes.
अब
सर एक बार एक्सप्लेन करो ना.
क्या? Who's there? Speak something clear.
Sir, I am Sharon. I just said, please, can you explain it again?
Okay, I said that whenever plant has to grow, there will be two levels of growth: primary growth and secondary growth. Up to maturity, primary growth, and even then after, if it continues to grow, then it is called as secondary growth. Seen in dicot, both of them, but in monocot only primary growth. आगे जाते हैं. When a primary growth occurs, whatever tissues are formed are known as primary tissues. During the secondary growth, whatever tissues are formed are known as secondary tissues. फिर आगे. This includes different types of tissues. उसमें से है primary xylem and primary phloem. इसमें भी secondary xylem form होता है and secondary phloem form होता है. Up to here, the things must be clear on your side. Are they clear?
Yes, sir.
I am explaining
Sir.
this. कौन है भाई?
સર
हेलो. हाँ.
સર મને એવું પૂછવું હતું કે જીમ્નોસ્પર્મ્સમાં સેકન્ડરી ગ્રોથ થાય કારણ કે મોનોકોટ્સમાં તો નથી થતું.
Gymnosperms do show secondary growth. Yes, they do.
તો
Huh.
monocots it doesn't happen.
What?
Only in monocot it does not take place.
Huh. Exceptionally, there are few monocots also which show the secondary growth. Very few. So you can't say that complete 100% monocots do not show it, but you can say that with few exceptions, majority of the monocots do not show the secondary growth. There may be few exceptions which show, but majority of the monocots do not show the secondary growth. That is the line one has to remember. चलिए, आप लोगों को मुझे इस पॉइंट पे लाना था कि व्हाट इज अ प्राइमरी xylem. Primary xylem is the xylem tissue developed during the primary growth of the plant organ. I go ahead now to the next. कौन है भाई?
સર શ્રેયા.
हाँ बोलिए.
Sir, do pteridophytes also have these vascular tissues?
Definitely I have taught this thing. Tracheophytes का पहला ग्रुप टेरिडो, दूसरा जिम्नो और तीसरा एंजियो था. They do have.
Okay, sir. Thank you.
अच्छा. This primary xylem. Further, I am explaining the primary xylem. Since the plant is in the form of a sapling, till it becomes a mature, whatever components of xylem are added, they are known as primary xylem. Okay. In that also two possibilities are there. Number one, that xylem when it is earlier formed in the early part of the life of that plant, sapling born हुआ, फिर थोड़ा smaller plant के form में mature हुआ. So that stage it is known as protoxylem. और थोड़ा मेच्योरिटी की टुवर्ड्स जब वो डेवलप होने लगा, उसके बाद जो xylem ऐड होता है, so the protoxylem, primary xylem added later on in the life of a primary growth of the plant, that is termed as metaxylem. आप इतना अंडरस्टैंड कर लो ना कि एक प्लांट के प्राइमरी ग्रोथ के अर्ली फेज में जो xylem डेवलप होता है, दैट इज अ प्रोटोक्साइलम. लेटर फेज में जो डेवलप होता है वो मेटाक्साइलम. फिर उसके बाद प्राइमरी ग्रोथ ओवर. So this is supposed to be the earlier form. पहले फॉर्म होता है तो वो थोड़ा नैरो होता है, उसके ट्यूब्स, xylem के वेसल्स के ट्यूब्स की डायमीटर नैरो होती है. This is formed later on and this is comparatively having wider constituents in it. इसके constituents थोड़े wider होते हैं. अब मैं आपको एक डायग्राम के थ्रू ये एक्सप्लेन करता हूं, आप इसको अंडरस्टैंड करिएगा. Let's look at this diagram. Let's imagine that this is the development of a root. ये root था, वो maturity जैसे सैपलिंग से पूरे मैच्योरिटी तक का उसका प्राइमरी ग्रोथ ऑफ द रूट इज शोन ओवर हियर. Here you can make out that initially formed xylem units are narrow. These units are called as protoxylem. These are nothing but the vessels of the xylem, but we call them as the protoxylem vessels. Kyun? Because they are developed in the early stages of the life of the plant. They are narrow in diameter. अच्छा, प्लांट थोड़ा मैच्योर हो जाता है, रूट की साइज बढ़ जाती है, newly added vessels will be broader in diameter. So these are going to be known as which xylem? Metaxylem vessels, isn't it? So you have two types of vessels. These which are encircled are which vessels? Metaxylem vessels. And these which are on periphery are which vessels? Protoxylem vessels. How many have understood this idea of protoxylem and metaxylem? Protoxylem and metaxylem vessels over here. बोलिए.
Yes, sir.
प्राइमरी xylem के दो फॉर्मेट है ये. एक तो आपको पता चलना चाहिए कि प्राइमरी xylem क्या होता है, सेकेंडरी xylem क्या होता है. सो द xylem constituents ये सभी मिलके सभी है कौन से वाले? प्राइमरी xylem. क्यों? Because they all are developed during primary growth before the secondary growth started. So we call them as primary xylem. अच्छा, in that those primary xylem constituents which were developed earlier in the life of plant, their origin is earlier and they have a narrow diameter क्योंकि वो पहले ऐड हुए थे. जब रूट की थिकनेस खुद इतनी कम थी तो उसमें ऐड होने वाले xylem vessels भी नैरो होते हैं. जब रूट की थिकनेस बढ़ गई है तो इसमें ऐड होने वाले वेसल्स बिगर होने चाहिए. कॉमन सेंस की मैटर है. सो बेसिकली the earlier added xylem vessels are narrow. They are called protoxylem vessels, those which are encircled in blue. And later added xylem vessels are broader. They are later and broader. इतना याद रखने का. They are called as metaxylem. They are known as what? Metaxylem. Protoxylem पहले आता है, metaxylem बाद में आता है. So that's how this arrangement is there. Are you all clear with the term protoxylem and metaxylem? दो वर्ड में अगर प्रोटोक्साइलम को डिफाइन करना है, दो वर्ड्स सिर्फ लिखना है उसके बारे में, तो कौन से दो वर्ड्स लिखेंगे? Which two words you will quote in order to mention protoxylem features? Earlier and narrow. Primary is wrong, Jugal, because even metaxylem is primary. Origin is earlier, diameter is narrow. That is about the protoxylem vessels. Two terms for metaxylem vessels. Later and wider. Origin is later, they are developed later on, and diameter is wider. That is about the protoxylem, metaxylem. I take you to the next level. The pattern of their development. Look, origin, timing point of view, one could be proto and another could be meta. I'm not talking about that. I'm talking about the direction in which they are developing within the organ. Direction का meaning दो possibility. Organ cylindricals है, sectional view spherical है, round है. So either the development of this tissue is taking place from periphery to center or from center to periphery. Based on that, first one, centripetal arrangement, the exarch development of xylem, that is from periphery to center, from periphery to center. Tell me, all of you, in this case, exarch development of xylem, which is normally seen in a root. Is it from periphery to center or from center to periphery? The development of xylem is taking place in which direction? Tell me. Exactly. It is from periphery to center. Can you justify? How can you confirm that it is from periphery to center? कोई है आप में से एक स्टूडेंट जो जस्टिफाई कर सके कि हाँ सर ये पेरिफेरी टू सेंटर का मैं एक्सप्लेनेशन दे सकता हूँ या दे सकती हूँ?
Hello,
बोलो.
sir. We saw that protoxylem are earlier and narrow. So here in the figure we can see that the protoxylem are narrow at the periphery and wider in the central portion. So
हाँ,
periphery.
दिस इज द रूट इन द अर्ली स्टेजेस. ये फिर थोड़ा और मैच्योर रूट है, ये थोड़ा और मैच्योर रूट है, ये इसमें मोस्ट मैच्योर्ड रूट है. देखो पहले कहां पे आया प्रोटोक्साइलम? पेरिफेरी पे. फिर metaxylem towards which position it came in a mature plant? It came towards the center. So the development of the xylem is from periphery to center. This is called as exarch development of the, you know, exarch development of the xylem. Simple as that, particularly primary xylem. अच्छा, ये अगर वास्कुलर बंडल है स्टेम का, तो इन दैट इफ यू सी के एक सेक्शनल व्यू अगर लिया जाए स्टेम का और उसमें ऐसे बंडल्स होते हैं, तो ये सेंट्रल रीजन है स्टेम का और ये पेरिफेरल रीजन. तो इन दैट इफ यू सी द डेवलपमेंट इज फ्रॉम सेंटर टू पेरिफेरी. फ्रॉम सेंटर टू पेरिफेरी. देखो प्रोटोक्साइलम, फिर मेटाक्साइलम. This part will be towards periphery, this part will be towards center. So as you can make out, the development is from center to periphery in case of a stem. जब root xylem develop होता है तो वो exarch pattern में develop होता है, that is from periphery to center, whereas the stem xylem, as in when it develops from center to periphery. It is called as endarch with some diagram. First is centripetal and another one is centrifugal respectively. Exarch and endarch. With some diagram, if I can justify this thing, have a look at this one. This is the section of a root. And this is the TS transverse section of a stem. This is the periphery of these organs, and these are the centers of these organs, central regions. Now you can see that it is the exarch arrangement from periphery to center. Obviously यहां से नहीं, यहां से यहां पे आपको एक्चुअली देखना है, और यहां पे from center to periphery. So this is the endarch development of xylem. Compare and understand the both. If you have understood, then only you write down yes, it is understood. Otherwise do not write down, no reply. Only those who have understood should reply in form of yes or clear. 2 students, 4, 6, 8, 10, 12 have understood so far, 14 have understood, 16 Aneri Kushi, Mahavir, Muhammad Mutahir Darogaji, 18 students have understood this thing. How about other 32 or almost 30 students over there? Have you all understood or not? Okay, it seems that at least majority have understood. So I have completed this topic of exarch and endarch development. Excuse me. As I mentioned, the xylem that develops earlier is called as protoxylem. The xylem that develops later on is called as metaxylem. During the exarch development, the centripetal development, the protoxylem will be placed towards periphery, metaxylem will be placed towards center. This is exarch development. It is to be seen in root. In case of endarch development, the protoxylem shall be placed towards center, metaxylem shall be placed towards periphery, and it will be seen in stem. We have completed the detailed study of the topic, that is the conducting tissues. Our next topic is vascular bundles, which is related with the conducting tissues over there. Vascular bundles over there. See, in a plant body, the xylem and phloem, the conducting tissues, more or less they are arranged towards the central region. Rather than periphery, more or less they are towards central region, and they get organized into groups, bundles. We call these units of conducting tissue such as xylem and phloem as vascular bundles. Is it clear to you what are vascular bundle? The conducting tissues such as xylem and phloem, they tend to remain arranged in the form of units, groups that are known as vascular bundles over there. Tell me, is it understood to you? Fine, you said yes. I will show you the pics to make you understand what I am talking about. जैसे अगर ये रूट का सेक्शन है, ये रूट था, उसका सेक्शन लिया हमने, ये सेक्शन का एक छोटा सा पार्ट है. This part is having vascular bundles. यहाँ पे phloem की अरेंजमेंट है, यहाँ पे xylem की अरेंजमेंट है. This is the region with vascular bundles over there. दूसरा डायग्राम, दूसरा पिक. Look at this section of a stem. स्टेम का बड़ा सा सेक्शन है, उसमें से एक छोटा पार्ट हम ऑब्जर्व कर रहे हैं. वहां पे ये एक वास्कुलर बंडल है, ये एक वास्कुलर बंडल है. स्टेम में भी होते हैं वास्कुलर बंडल्स. Check out this leaf also. In this leaf also, if you see carefully, vascular bundles are present over there. Xylem and phloem making the vascular bundle in this region also. So by and large, you can make out that whether it is the root, stem, or leaf, the xylem and phloem are organized in them in this format as a units over there. We call these units as vascular bundles. These vascular bundles are categorized into two major categories. One is called radial bundles and another one is called conjoint bundles. इसको एक कैटेगरी कहते हैं, इसको दूसरी कैटेगरी कहते हैं. दो मेजर कैटेगरीज के होते हैं बंडल्स, VBs. I would be calling them as the VBs in short. V for vascular, B for bundles. VBs, they are of two categories. One category is called as radial, another category is called as conjoint. See, this is one bundle. This is another bundle. Here, this itself is a one bundle, vascular bundle. If you compare these bundles, each one has got phloem and xylem, phloem and xylem, phloem and xylem, phloem and xylem, phloem and xylem. In this case, if you see, either the bundle is only phloem bundle or only xylem bundle. This is the main difference between these two categories of bundles, what you call as radial bundles and conjoint bundles. How many have understood? A given vascular bundle, if it is made up of only xylem or only phloem, it is a radial type of bundle. Xylem bundles are separate and phloem bundles are separate, as simple as that. In case of a conjoint type of a vascular bundle, within the same vascular bundle, the xylem and phloem are present side by side. We call it as a conjoint type of vascular bundle. That's the first difference between radial and conjoint. अच्छा, रेडियल को डिटेल में कोई स्टडी करता है तो इट विल बी लाइक दिस, कि जैसे ये ऑर्गन का सेक्शनल व्यू है और ये ऑर्गन का सेंटर है. सो यू कैन मेक आउट दैट on the different radii, radius का plural होता है radii. So can you make out that xylem and phloem bundles are separate? Two, they occur on separate radii. अगर इस रेडियस पे xylem है तो इस वाले पे phloem है. अगेन इस रेडियस पे xylem अरेंज है तो यहाँ पे phloem. Xylem, phloem, xylem and phloem. So number one, xylem bundles and phloem bundles are separate, or you can say xylem bundles are separate, phloem bundles are separate. Two, they occur on separate radii. Three, they occur alternate to each other. Four, they are found in the root. चलो, अगर किसी को ये 4 पॉइंट्स क्लियर हो गए हैं तो हैंड रेस करो या फिर क्लियर लिखो या फिर यस लिखो. अगर किसी को डाउट हुआ है तो फिर नॉट क्लियर लिखो, क्वेश्चन पूछो, क्लैरिफाई करो. देखो, जैसे ये एक बंडल है, वैसे ये भी एक बंडल है, ये भी एक बंडल है, ये भी एक बंडल है. इस बंडल्स को आप कंपेयर करोगे तो गिवन बंडल में है ओनली xylem और ओनली phloem इन इट, बट इस बंडल में xylem भी है और phloem भी है, दोनों होते हैं. नंबर वन. सो देयर इज अ फर्स्ट डिफरेंस. Radial bundles are made up of only xylem or else only phloem alone, not together in a same bundle. Second, generally these bundles tend to remain on a separate radii, and that too in an alternate manner where xylem bundle is followed by phloem. Like that, the arrangement is there. This type of pattern is seen in the bundles of root. Example, monocot या dicot root का sectional view है ये. अगर तो यहाँ पे दोनों में ऐसा दिखेगा कि ये रेडियल बंडल्स दिखेंगे दोनों में, मोनोकोट या डाइकोट के रूट में बेसिकली. You can see that this is the bundle of xylem and these are the bundles of phloem. And as you can see carefully that each one is on a separate radius. रेडियल बंडल के बारे में 4 लाइन अगर क्लियर हो गया, 4 लाइन्स क्लियर अबाउट रेडियल बंडल, देन वी कैन गो टू द नेक्स्ट वन. Point 1 लिखो. Fatima, can you speak about 4 lines quickly so we can go to the next?
Yes, sir.
1.
In radial bundle, either xylem or phloem are present. They are on the separated radius, and xylem is followed by phloem, and they are
alternate arrangement we say.
yes, sir, and they are in roots. They are found in roots.
Yes, number 1. The given vascular bundle may contain either xylem or phloem. So xylem and phloem bundles are separate. 2. On separate radii. 3. Alternate to each other. 4. Such arrangement is seen in case of root. Next one. The other category is the conjoint. Can you say in one line what is the speciality of conjoint bundle that is commonly seen in all of them? Yeah, within a same bundle, one can see the xylem and phloem together. Acha, in that, one possibility is that it can be concentric, it can be collateral, it can be bicollateral. All this under conjoint. So all of them shall have phloem and xylem together in a same bundle. But for example, if we discuss about the concentric pattern, the one in which, as you can see, one type of conducting tissue encircles other. जैसे एम्फी वेसल में xylem encircles phloem. In amphicribal, phloem encircles xylem. Is it visible to you, this arrangement? That conjoint pattern in which not only they are together, but one of them encircles the other. The xylem encircles the phloem in case of amphivessel, and phloem encircles the xylem in amphicribal. Both come under concentric type of conjoint bundles. Examples are quoted below them. How many are clear with the concentric type in amphivessel and amphicribal, respectively in Euca stem and fern stem? If it is not clear, at least you have to let us know that you are not clear. Oh, it seems it is clear. Done. Second category, collateral. Second category is which one? Collateral. Yucca plant में amphivessel and fern plant में amphicribal. These are the examples of concentric type of VB showing plants. We move to the next one that is called collateral. Acha, can you see that in collateral in a given bundle, both xylem phloem are there? So both are the examples of conjoint. But unlike the concentric, where one encircles the other completely, here is that they occur side by side without encircling each other. Normally phloem on outer side in the bundle and xylem on inner side of the bundle. That is called collateral category of VB. In collateral category of VB, you can see the xylem phloem together in a same bundle side by side, phloem on outer side and xylem on inner side on the same radius. That is called as collateral type of bundle. In this also there are two patterns prevalent. The one in which between the xylem and phloem there is no meristematic cambium tissue. The vascular cambium strip is absent. It is called as closed type of conjoint bundle. Example, monocot stems, maize, etc. That collateral bundle in which between the xylem and phloem within the bundle, the vascular cambium is passing on outer side of the cambium phloem, on inner side of the cambium there is a xylem. This pattern is called as open type of collateral bundle. Open type of collateral bundle. It is normally seen in dicot stem. देखो रूट में एक ही ऑप्शन रेडियल, स्टेम में मोनोकॉट, स्टेम में closed bundles, dicot stem में open bundles. That's the way one has to understand. Last, bicollateral. As you can see that in the same bundle, you have a xylem and phloem together, but actually phloem two patches, one on outer side and one on inner side, and between each patch of phloem and xylem there is a cambium tissue. ऑल अराउंड होता तो ये एम्फिक्रिबल बन जाता, फ्लोएम ऑल अराउंड द xylem होता तो एम्फिक्रिबल हो जाता, एक साइड पे होता ऑन आउटर साइड तो ये कोलैटरल हो जाता, दोनों साइड पे है तो ये कौन सा है, बाइकोलैटरल कैटेगरी है. दिस इज द बाइकोलैटरल कैटेगरी ऑफ द VB, द गिवन VB. Now if I show you in this pics, first of all dicot root, which category, which category of vascular bundles are these? जिसको मैं येलो से आंसर करूंगा वो vascular bundles हो गए. Which category are these vascular bundles? Radial. Another example. Monocot root. Vascular bundles. Which category? These are vascular bundles. Again, which one? Radial. Now comes dicot stem. These vascular bundle. ये cambium, ये phloem और ये xylem. Tell me, which category is this? Open collateral conjoint. Conjoint because xylem phloem together. Collateral because phloem only on one side of the xylem. And open because between the phloem and xylem there is a cambium tissue. Another one, monocot stem. This is the vascular bundle. This is the phloem. These are the xylem constituents. No cambium in between. Which three terms you can employ for the same? This type of VB. Conjoint collateral closed. If the previous one was CCO, conjoint collateral open. This is CCC, conjoint collateral closed category of bundle. क्यों conjoint? एक word दे सकते हो conjoint? उसके लिए एक line बोल सकते हो? What do you mean by conjoint? Xylem phloem together in a same bundle. Jayati, correct. Next one, can you explain why collateral? Collateral का meaning क्या होता है? Collateral, side by side, where phloem is on outer side on the same radius to that of xylem, isn't it? And what about closed? Closed term is for which reason? No cambium within the bundle. See, this is which category? Radial. Reason? Xylem bundles are separate. Phloem bundles are separate. They are on different radii and are alternate to each other. This is to be seen in root. Simple as that. Okay, done. Same you have seen in this symbolic diagram. चलिए ये वाला रेडियल है या कॉन्जॉइंट है? नंबर 1, this bundle is radial or conjoint because xylem and phloem are together. Okay. दूसरा, concentric है, collateral है या bicollateral? Concentric, because one tissue is completely encircling the other. You can make out in this too. जो मैंने एनसर्कल किया है रेड से, वो एम्फिक्रिबल है या एम्फी वेसल है? Xylem outside and phloem inside. It should be amphi vessel. Amphi vessel होना चाहिए. And the one which is encircled with the blue, this vascular bundle, where phloem is on outer side and xylem is on inner side. It is amphi. This next one, H, G and H are which one? The blue one encircled with amphicribal over there. Amphi vessel in Yucca, Dracaena, etc. plants. Amphicribal in Fern, Selaginella type of pteridophytes. Tell me, ये bundle, अच्छा ये bundle, the one which is encircled with yellow, कौन सी type का है? Radial या conjoint? करेक्ट. कॉन्जॉइंट. कॉन्जॉइंट में कॉन्सेंट्रिक, कोलेट्रल या बाइकोलेट्रल? कोलेट्रल. करेक्ट. कोलेट्रल में ओपन या क्लोज्ड? ओपन. टू बी सीन इन डाइकोट और मोनोकोट स्टेम? डाइकोट स्टेम, माइंड यू. दूसरा वाला, दिस बंडल. इसके लिए 3 वर्ड साथ में लिखने हो तो कौन सा 3 का कॉम्बी लिखेंगे? हाँ, conjoint collateral closed. Without cambium between phloem and xylem, it's conjoint collateral closed. किसमें दिखता है? मोनोकोट रूट में या डाइकोट रूट में? पूरा लिखना. रूट या स्टेम या, देखो रुद्र ने मोनोकोट रूट लिखा. I wanted the same mistake to happen from your side. I said radial arrangement in the root, so how come the root is the answer in this case? Stem होना चाहिए ना आंसर. It is to be seen in monocot stem. Remember this thing. And this last one, when I point out this last one, phloem, outer phloem, inner phloem, outer cambium, inner cambium, and xylem. Which is this category? Which is this category? Yeah, bicollateral, conjoint, and bicollateral. Example, cucurbita, कुकुम्बर और उन सब सिमिलर प्लांट्स के स्टेम में ये अरेंजमेंट होता है. Well, here I conclude the topic of vascular bundle and their types. Again, I give you an opportunity to ask a question in case if you have any. अगर आपको कोई भी क्वेश्चन या डाउट है तो आप पूछ सकते हो. No, it seems you have no questions, no doubts. That is amazing. कुछ रिवाइज करना है, कोई क्लैरिटी चाहिए, कोई एग्जाम्पल, कोई डाउट, कुछ पूछना है तो हाँ. As a homework, you have to draw all these diagrams, the one which are there on the screen. As it is, chart पूरा draw करना है आपको. Xylem phloem के जो एक टेक्स्ट बुक वाले डायग्राम है वो भी ड्रॉ करने हैं. पैरेन्काइमा, कोलेन्काइमा, स्क्लेरेन्काइमा के डायग्राम्स एंड इवन मेरिस्टेमेटिक टिश्यूज के. Whatever diagrams are given in your textbook, same diagrams must be prepared by you. That is the homework which we will check in our next lecture. I guess I made it very clear now what homework is given to you. Is it clear to all? देखो अभी एक लास्ट इंस्ट्रक्शन and then we wind up. See, the logic of having the recorded video is that on the day when you learn the topic, almost on the same day or within 48 hours, go through that video again to understand the topic. Later on, when exams appear, at that time, actually, this is not required to go through the lectures, because if you have in a chapter 9, for example, if there are 6 or 7 lectures, so almost 9 hours matter. You just for 9 hours, you go through those videos only. Why do you get over dependent upon the technology? You have understood the topic in a running lecture, then after you revise it at home immediately within 48 hours by our recorded video. And then as and when test appears over there, you don't need to go through the videos again. There you need to go through the textbook. You have to read the textbook. You people do not read the textbook and you get a lesser marks. Believe me, the students who are getting 20 plus marks will get less than 15 marks if there is a physical examination of your papers. And if you write in front of us, I bet, I challenge to you, very few can cross 20 marks. हमारा सेट किया हुआ पेपर हमारे सामने लिखो और हम उसकी आपके सामने assessment करेंगे checking का, तो you won't get even 20 marks. So these are not your real marks actually. Reason is that you have no habit of writing and reading. आपको सिर्फ लेक्चर सुनना अच्छा लगता है तो बाद में भी रिवाइज में भी वो लेक्चर सुनते हो. सुनने से नहीं लिख सकते हो, रीड करने से, राइट करने से लिख सकते हो. एक एजुकेटेड स्टूडेंट अप टू 10th स्टैंडर्ड, उसको इतनी समझ आ जानी चाहिए. The thing which I want to convey, that you cannot remember things by just listening. Listening is one of the way, not the only way. Research has proven that if there are multiple sources by which the information comes in your brain, through hearing as well as through seeing as well as through reading and writing, when you are practicing the thing, then your content is becoming stronger. Is there anyone who feels confident that, sir, I can write in front of you your paper and get 25 out of 25 or 22 out of 25? I can bet you cannot cross 20. Is there anyone? So you know it very well that it is wrong thing that you are doing on your part. You are misleading your parents and teachers also and yourself, thinking that listening these videos will help you to get a good score. It can never actually. You don't know the logic of revision. हाँ, अगर नीट एग्जाम 2 years के बाद आता है और उस टाइम पे आप ये वीडियोस देखते हो तो आपको पूरा एक बार रिवाइज हो सकता है. अभी आपने लेक्चर में पढ़ा है 2 week पहले, फिर अभी इट्स नॉट फॉर वन स्टूडेंट, आई एम टॉकिंग फॉर ऑल. अभी आपने लेक्चर में पढ़ा था, उसके 2 दिन बाद वीडियो में भी देखा होगा, बिकॉज इट वाज रिलीज्ड विद इन अ डे, तो वो 48 hours में भी देखा होगा. और अब भी आप उसको देखोगे, व्हाट इज द सेंस? कॉमन सेंस वाला पर्सन इसको फिर से नहीं देखना चाहेगा. And this is for all 48 of you. All of you are alike. ये वीडियोस लॉजिकलेस है, अगर इनको बार बार देखना. इनका टाइमिंग इंपॉर्टेंट है. रीडिंग किया है और उसके बाद कोई डाउट है तो एक बार देख लो, तो वो 48 hours में ये सब एक्सरसाइज कंप्लीट हो जाना चाहिए. फिर आप और आपकी टेक्स्ट बुक या आपकी नोटबुक, दैट इज द थिंग विच इज लेफ्ट फॉर द टेस्ट प्रिपरेशन. I hope I have conveyed it correctly. My tone may be little bitter or stronger, but message that I am conveying is better for you in overall preparatory arrangement. अगर आपको अच्छा लगा तो ठीक है, otherwise we shall be providing the videos. You can definitely go through that. I conclude the meet.